Hello, Anatomy and Physiology students. This is Dr. Bob here. And the topic that we're going to be discussing next is muscular tissue. And as you see, MMA professional Georges St. Pierre is showing some of his muscles to kind of get us started. So the first question we have to ask is, what do we use muscles for? Hey, number one, muscles are used for movement. Now, what do we move with muscles? Well, we move the different parts of our body. We move appendages. We move blood when our cardiac muscle in our heart pumps blood. We move food through our digestive tract or our alimentary canal as we are taking the nutrients from that. So muscles are going to be used for movement. Muscles are also used to help maintain body position and posture. It is through the contraction of these muscles that we are able to be in an upright position and do the stuff that we need to do. Muscles are going to play an important role in helping to support and protect some soft tissues. Now, yes, we do have our skeletal system that provides protection for things like our brain. But if you look at the abdominal muscles on the anterior portion of our body, they're going to be providing protection from impacts like these martial artists that you see right here getting kicked in the ab muscles. Were it not for strong abdominal musculature, boy, there could be some serious damage that can be done. Next thing that we use muscles for is to help control entrances and exits to things. There are muscles called sphincters, and these sphincters are circular muscles, so they'll go around an organ or a structure, and by contracting, they cause those structures to get smaller. So in the urinary bladder, which what we see here, is going to store urine, and the urine would then move out of the body through the urethra. There are two sets of sphincter muscles. You'll learn this in 245, the internal urethral sphincter and the external urethral sphincter. And it's by contracting these muscles that we prevent the urine from leaving the body when we don't want it to. Muscle's going to play an important role in helping to maintain body temperature, because if you think back to your general biology class, remember when we talked about aerobic respiration? Okay, converting energy from one form to another, so taking the energy in a glucose molecule and using it to make ATP, and then taking ATP and bringing it down to release the energy for the cells to do the stuff that they need to do. You may remember that when that happens, that some of that energy is lost as heat. And the heat from the contraction of our muscles is going to allow us to do temperature homeostasis. All right, so let's look at some of the characteristics of muscle tissue. So muscles have what we call electrical excitability. Woo! No, not that kind of excitability. Electrical excitability means that they can move action potentials, which was the last topic that we talked about. Muscles, interestingly, have the ability to contract, and the term for that is contractility. So muscles are the only cells in our body that have the ability to make themselves smaller. So if you look at the biceps brachii of this woman right here, the muscle would be located on the anterior portion of her arm. When the appropriate action potentials are sent to the biceps brachii, it's going to cause these muscles to get shorter, and by shortening the muscles of the biceps brachii, we're going to make the angle here at the elbow smaller. So if we see our straight line here is 180 degrees, when she contracts her biceps brachii muscle, it's going to cause flexion. We're going to go from 180 degrees right here to, that's probably about 45 degrees at the angle here. So making the angle smaller is called flexion. So by flexing her arm at the elbow using the biceps brachii muscle, she's demonstrating that the biceps brachii and all of our muscles have contractility. Now, not only can we contract muscles, and that's good, but if all we can do is contract muscles once and then not do anything else, well, that doesn't help us very much. So muscles also have extensibility, meaning that after we have contracted them, they can be stretched back out to their original length. So if here was her resting state, She was contracting her biceps brachii, it's inflection. We can also have extension, or having her go back to this state right here where her arm is straight. Now, interestingly, muscles can't extend themselves. So one of the reasons why I chose this picture is you can see the young lady here is holding a dumbbell here, some hand weights. So by relaxing the biceps brachii, gravity is going to help extend the muscle. You can also have an antagonistic muscle doing that. And on the posterior side of the arm is the triceps brachii muscle. And by contracting the triceps brachii muscle, that will also cause extension, making the arm go back to the fully extended position that we see right here. So muscles cannot extend themselves. Muscles also have what we call elasticity, and it's pretty much, as the name implies, they can be kind of elastic. So if you stretch a muscle a little bit, like with this rubber band, here you see the rubber band here is being stretched. When you stop stretching, it can go back to its original length. So that's elasticity here. Now be careful. Muscles don't have the unlimited ability to show elasticity. They can only be stretched a certain amount, but they do go back to their original size afterwards. So the next thing we're going to talk about is we're going to organize the different levels of muscle complexity. And this is going to be important because we're going to be looking at muscles as a whole, and we're going to be looking at muscles at the microscopic level, and actually even pretty high magnification, so really small stuff. So I'm going to encourage you, when you're writing out these notes, and you should really be writing this stuff out, to write out the concept map that I'm going to be sharing with you here. So if you need to pause the video for a moment and get a pen and a pencil, please do so. In addition, if I go through this too fast, by all means, please hit pause and give yourself a chance to get caught up. But I will be going through this a little bit more quickly than I would in a regular class because you have the ability to pause when you need to. So we're going to start with our biggest structure, which is a muscle, like perhaps the biceps brachii, or any of the other muscles that we'll be learning about in the lab. Okay, a subdivision of a muscle is what we call a fascicle. A whole bunch of fascicles together are going to make up a muscle. Subdivision of a fascicle is going to be a muscle fiber. And please keep in mind that we're talking about skeletal muscle here. We're not talking about smooth muscle and cardiac muscle. Those are going to be arranged a little differently. But honestly, if you're thinking about taking the NLN exam or the MCAT for med school or any of a variety of other tests, standard practice is to talk about what's going on in skeletal muscle. So we'll stick with that standard practice. Within muscle fibers, they're cells, right? So they've got lots of different structures. But the three structures that are going to be important to us in order to understand muscle physiology, what happens when a muscle contracts, are the sarcoplasmic reticulum, the sarcoplasm, and the sarcolemma. And we will talk about all of the structures in this concept map in this video today. Also within a muscle fiber, we're going to have many myofibrils. And the myofibrils we're going to see are organized into sarcomeres. And again, don't worry if you're not getting all the terms. We will be going through all of this stuff in this video and the next, so stay tuned. Within each myofibril, we're going to have the myofilaments. And sometimes the myofilaments are just called the proteins. And the two myofilaments that we're going to be talking about are actin proteins or actin myofilaments and myosin proteins, myosin myofilaments. And within each one of these, we're going to have structures. So in actin, we're going to see F-actin, tropomyosin, and troponin. And in myosin, we're going to see the head and the tail. So hopefully you see from this, we see a lot of terms that begin with sarco and a lot of terms that begin with myo. And both of these are going to be pertaining to muscle. So get used to them. We're going to see a pretty fair number. That's also why we go through a concept map like this, is to help you keep track of all the different terms, many of which are going to be sounding alike. So the parts of a skeletal muscle is what we're going to talk about next. So a muscle, whether we're talking about the biceps brachii or any of the abdominal muscles or any of the skeletal muscles that we're going to be talking about, are made of many muscle fibers. And these muscle fibers are going to have a variety of 3 connective tissues that are working with them to help give the muscle its shape and to allow the muscle to do its job. The most superficial of those connective tissues is what's called epimysium, or sometimes it's called fascia. This is a connective tissue that's superficial to the entire muscle. And in fact, if you think back to when we talked about the skeletal system, we said periosteum is the connective tissue that's going to be superficial to bone. And we said the tendon is going to be a structure that connects a muscle to the bone, and that tendon actually grows into the periosteum. Well, all 3 of these connective tissue layers are going to help form the tendon and grow into the periosteum of the bone. So that's going to give us a really, really strong connection. And the most superficial of that layer is going to be the epimysium. Deep to the epimysium is going to be the perimysium. And the perimysium is going to be this connective tissue layer that will surround individual fascicles. And then the deepest of all of these connective tissue layers is going to be called the endomysium. And the endomysium is going to surround each muscle fiber. So endomysium, perimysium, and epimysium, all these connective tissues that are associated with the muscles actually grow together. And when they grow together, they form the tendon that actually grows into the periosteum. And it's these layers that are going to form the tendon that connects the muscle to the bone. So if we take a look, you got a couple of different things going on here. So here is obviously a bone. Here's a tendon that is going to be connecting to a muscle, and this muscle was cut in cross section so that we can see it more clearly. So this whole thing here would be a muscle. Notice within the muscle we've got a packet of stuff here, we've got a packet of stuff here, and a packet of stuff here. Here is one of those packets that actually been pulled out a little bit. So you can see the different components of the fascicle with the perimysium that would be around this. And then you can see individual muscle fibers. Now these would be microscopic, right? So this isn't really true life size. But this would also be surrounded by connective tissue, the endomysium. And it's all 3 of these tissues, the epimysium, the perimysium, and the endomysium, that grow together to form the tendon, which connects the muscle to the bone. And I included this picture from a forearm model because you can see some of these tendons. Like this is the muscle we'll be learning about in class called the flexor carpi radialis. So here's the flexor carpi radialis tendon. You can see it moving along the forearm here. It's really quite obvious. So this brings us to the word fascicle. All right, so a fascicle is a subdivision of a muscle. Each fascicle is going to contain many muscle fibers. And a muscle fiber, you should remember this from when we talked about histology in the beginning of the semester, is an extremely long multinucleated, and I use the word cell in parentheses, right? Because really what happened during embryonic development, those skeletal muscle cells kind of grew together and they fused, giving us this great big long, we can't really truly call it a cell because it's got multiple nuclei along its length. But the muscle fiber, is similar to the cell that we would see in cardiac muscle or in smooth muscle. These are among the biggest cells in the body. Each muscle fiber can be up to around 12 inches or 30 centimeters long. And as I said, they form from the fusion of embryonic myoblasts. Now, here's a word we haven't seen before, myoblasts. Do we remember what blast means? Well, hopefully you remember, think B, blast, build. Myo means muscle, so myoblasts are the muscle building cells that would be forming in an embryo to give us our skeletal muscle fibers. And here you can see a skeletal muscle, actually 3 skeletal muscle fibers. So there's one going to be here, there's one here, There's another one here, and notice the multiple nuclei that you find along the length. Sarcolemma is an important concept for us to talk about, and the sarcolemma represents the cell membrane of a muscle fiber. And if you remember from your general biology class, we've got the cell membrane being a phospholipid bilayer with proteins embedded into the surface, and that's all true. But the sarcolemma of a muscle fiber is going to have something that most other cell membranes don't have. And that's going to be something called a T-tubule. And the letter T here means transverse. So within the sarcolemma of a muscle fiber, we have T-tubules or transverse tubules. And the definition for it is a tube-like invagination in the sarcolemma. What this means to us is if here you see the cell membrane of a cell, there's essentially this pinched-in area. And if you notice, this pinched-in area is going to go deep into the cell. Now, looking ahead a little bit, and applying some of what we learned from our action potential work. So an action potential is going to be traveling along the sarcolemma of a muscle fiber. The presence of these T-tubules allows the action potential to be delivered deep into the muscle fiber to the myofibrils that you can see one of right here. Don't worry, we'll talk more about that soon. So T-tubules are going to allow for action potentials to be delivered deep into the muscle fiber, which is a very good thing. Now I realize sometimes it's hard to visualize what these T-tubules are like, even though we have this picture here. So a visual I'll often use for my in-person classes is imagine you had a balloon. So here you see an orange balloon that I haven't blown up too terribly much. And I'm taking my thumb and I'm actually sticking my thumb into the balloon. And you notice how the balloon would wrap around your thumb, or if you're doing it with another finger, how it would wrap around your finger. That's really what the T-tubules are like. So the cell membrane, the sarcolemma, actually wraps around your thumb, and the way that it wraps around your thumb would be like the way that cell membrane dips deep into the muscle fiber. And that's important. Because what's an action potential? It is a wave of depolarization and repolarization traveling along the length of the cell membrane. So you have to have sarcolemma in the T-tubule there, because without cell membrane you couldn't have action potential. And remember that, because that's going to be important. Next part of a skeletal muscle fiber is something we call the sarcoplasm. And you should notice that sarcoplasm looks an awful lot like cytoplasm. Sarcoplasm is in fact the cytoplasm of a muscle fiber. And it contains a very important protein called myoglobin. There's that myo prefix again, meaning muscle. And like hemoglobin, myoglobin is a very similar protein whose job is to carry oxygen. Why does a skeletal muscle fiber need oxygen? Well, why do all cells need oxygen? For aerobic respiration. So you got to have oxygen to break down glucose in order to make ATP. Now, perhaps you've done some grilling and you take your steak and you season it up and you leave it on your plate for a little while. I was doing that this summer with some shish kebabs, and you notice you've got this thin red stuff at the bottom of your plate before you put it on the grill. That's not blood. Blood would be a lot thicker and it would be coagulating, it would be clotting. What you're seeing here is myoglobin. So when you cut into a steak, well, let's think of uncooked. So you're cooking into essentially cow muscle if you're doing steak. That fluid that's leaking out is actually cytoplasm or sarcoplasm from our skeletal muscle fiber that contains the myoglobin. You got to have that for binding oxygen, for making ATP. We also find within a muscle fiber structure called sarcoplasmic reticulum. And this word, again, seems an awful lot like a word you've learned before. Endoplasmic reticulum, with that prefix sarco here meaning muscle. So the sarcoplasmic reticulum is the smooth endoplasmic reticulum of a muscle fiber. And this is really, really important, and we're going to keep coming back to this important ion here. The sarcoplasmic reticulum stores calcium. Now, you may remember from our first exam that we talked about calcium balance, calcium homeostasis, and you got to have calcium for things like muscles to contract. Well, here we're seeing that calcium is going to play a very important role in muscle contractions. And calcium is stored when the cell or the muscle fiber is at rest in the sarcoplasmic reticulum. And we're going to see that again right here. So this kind of darker yellow, goldy kind of stuff on either side of our T-tubule. Here's a T-tubule. The stuff on either side of the T-tubule is sarcoplasmic reticulum. And we'll actually see the relationship between sarcoplasmic reticulum here, sarcoplasmic reticulum here, and T-tubule in between. We've got sarcoplasmic reticulum, T-tubule, sarcoplasmic reticulum. That gives us a structure with three things. So collectively, that's called a triad. So our triad is where you've got a T-tubule and two parts of the sarcoplasmic reticulum coming together. And you can see it even more clearly here. The next part of our skeletal muscle fiber represents myofibrils. Myofibrils are collections of proteins found within a muscle fiber. And you can see, this is a slightly different picture than the one we've seen before, but I like it because we can still see sarcolemma here, our cell membrane, and you can see quite clearly the T-tubules that would be going down in. But by removing the sarcolemma over here and then also cutting it in cross-section, you can see these bundles of tubes. These are the myofibrils. And they've even pulled one of the myofibrils out so you can see it more clearly. Keep in mind, this entire structure is a skeletal muscle fiber, right? So this is at the cellular level pretty small, much smaller than, say, the biceps brachii. Okay, so myofibrils are protein bundles that we find inside of a muscle fiber. And the myofibrils are going to contain structures called myofilaments. There we see another myo word. Myofibrils are the bigger ones inside of a muscle fiber here. The myofilaments are going to be the smaller things that we see inside the myofibrils. If you find yourself getting confused with all of these myo and sarco terms, go back and look at the concept map that we put up a little while ago, and that will help orient you onto what we're talking about. So we said there's two major types of myofilaments. We've got actin, which is the thin myofilament, and we've got myosin, which is the thick myofilament. If you have trouble remembering which is which, so the word actin has 1, 2, 3, 4, 5 letters. Word myosin has 1, 2, 3, 4, 5, 6. So the word with fewer letters is the thin myofilament. The word with more letters would be the thick. And there are other ways of remembering it, but sometimes quick and easy things like that is helpful. Why are we talking about these actin and myosin myofilaments? Well, we said one of the properties of skeletal muscle fibers and muscle fibers in general is that they contract. They can get smaller. And literally what's happening inside of our skeletal muscle fiber is because of the arrangement of actin and myosin, these proteins slide over one another. And it's that sliding over each other of those proteins that are going to allow the muscles to contract, get smaller, and allow them to be stretched back out to their original length too. And here's a picture that we'll be seeing multiple times as we go through these muscle notes. And a myofibril is made up of around 10,000 or so sarcomeres. So you know what we're going to talk about next, right? Got to talk about what is a sarcomere. So a sarcomere is the functional unit of a myofibril, meaning that if you could break a myofibril down into its smallest individual piece that would do its job of contracting, that would be our sarcomere. And remember, each myofibril is going to have around 10,000 or so of these sarcomeres, all of them contracting together at approximately the same time to make our entire myofibril and make our entire muscle fiber. And if we're contracting all of our muscle fibers, make all of our muscle contract. So the sarcomere is going to contain alternating dark bands, which are sometimes called A bands, and light bands, which are also called I bands. So when you look at this picture that you see right here, here we see a dark band and a light band, and a dark band and a light band. And hey, remember when we were talking about muscle histology, we said that one of the characteristics of skeletal muscle fibers is that they have these striations. And I said, don't use striations as a key characteristic because cardiac muscle cardiac muscle cells also have striations. But here's why we see striations. So it's these alternating dark bands and light bands and dark bands and light bands. And really what we're seeing are bundles of actin and myosin proteins that are organized in such a way to show us those dark bands or A bands and light bands or I bands. Now, if you are a kinesthetic learner, if you are a visual learner, even if you're a read-write learner, one of the best ways of learning the different parts of a sarcomere is to draw it. So remember when I told you a little earlier to have some paper and a pen or a pencil out? You're going to want to break that out now if you haven't already. And I would encourage you, if you have markers or crayons or something to do things in different colors, that's going to help as we go through this too. Please pause the video if you need to get those ready because we are drawing a sarcomere. Okay, welcome back. So I want you to start by drawing these big, thick, draw 3 of them, nice big vertical lines. And these will be the darkest lines that you draw. So if you've got a marker, draw them nice and thick. If you've got a crayon or a colored pencil, go back and fill them in and make them nice and thick. Okay, and again, if I'm going too fast, please pause the video, allow yourself to draw this, and then once you're done, please keep her moving. After you've drawn these big, thick vertical lines, which we will call M lines in a few minutes, I want you to draw some horizontal lines going through these. Now, before I go any further, I want to point something out to you. Actually, let me draw a few more of them. So I've got this organized this way on purpose. So notice there's an M line here, an M line here, and an M line here. Notice that these big, thick horizontal lines are not quite as thick as the M line here, but these thick horizontal lines are offset. So if I was to continue this line going this way, it would not touch up with one of these lines here. So they're supposed to be offset from one another. So as you draw these, make sure that you draw it in the same way and on both sides. Make sure that they're not one big line going all the way across. Once you've got these drawn, then we're going to start drawing some thin horizontal lines like this. Again, keep in mind that these thin horizontal lines that I'm drawing do not line up perfectly. They are kind of offset here. And that is also intentional because this is the way that you would find these protein fibers arranged in an actual sarcomere. Once you've got these lines drawn, then we're going to draw some zigzag lines that are going to connect up these thin myofilaments. Oh, I think I gave that away. And here we see there's a whole sarcomere here in between our zigzag lines that I've color-coded red. Color code them however you like. But we've got an entire sarcomere here, part of a sarcomere here, and another part of a sarcomere here. So let's look at what we've drawn. So our A band, or our dark band, and how convenient is this? They didn't do it this way, it just kind of worked out nicely. So what is the first vowel in the word dark? A. So our A band or dark band is where we see myosin, those thick horizontal lines, the thick myofilaments, even though there's also going to be actin there. And within our A band, we're going to have the M line. That was those very big green vertical lines, which are the first lines we drew. Within our dark band, we've got that M line, which is the central part of the A band, and it's going to contain stabilizing proteins. Now, I remember when I first learned this at the community college I went to. I was taking an anatomy and physiology class. I remember at this point feeling a little overwhelmed with A band or dark band and M line and H zone, which is coming up soon. If you're feeling yourself starting to get overwhelmed, stop the video, go back and review stuff, take a short break, and then go back to it, right? So there's a lot of definitions here, but with a little practice, you're going to get it. So in the middle of our A band or dark band, we've got the M line, and we've got the H zone, which is the area around the M line where we have the thick or myosin myofilaments, but we don't have any actin. I should probably put up a picture to show you some of this stuff. And I will after we talk about this next structure called the zone of overlap. And the zone of overlap is the part of the A band where both actin and myosin myofilaments overlap with one another. So here we see our picture. So this is almost identical to what we drew a little while ago. There's a dark band, there's a light band. There's a dark band, there's a light band. There's a dark band. I'm going to go do this again. Ready? So when we're looking at a sarcomere, it goes dark band and light band and dark band and light band and dark band, and next would be light band. All right, next. Notice the green rectangle I put on our area here. I'm going to take it away, and I'll put it back. So this represents our H zone. So when I take it away, here's our M line, smack dab in the middle of our dark band here. So surrounding the M line on either side, we've got this H zone. So these big blue horizontal lines are the thick or myosin myofilaments, and we know they overlap with the actins and through much of the dark band, but we've got this area here where we see myosin but no actin, and this area here where there's myosin and no actin. So we put that box back around it. That is our H zone, surrounding the big heavy green line here, which is our M line. And then we've got this yellow box that you see right here. This represents the zone of overlap because this is where we have both myosin, the thick myofilaments in blue, and the actin, or the thin myofilaments in black. And then we've got this area that I put the pink box around. This is our I band, which we'll be talking about soon. And smack dab in the middle of our I band, we've got this zigzag line here that's connecting up the actins of different sarcomeres. And we're going to call this zigzag line here in red the Z line or the Z disc. So let's talk about our I band. So our I band or light band, and again, notice what's the first vowel in the word light? It's I, which is awfully convenient. And the I band is where we're going to have actin, so our thin myofilaments, but no myosin, none of the thick. And it is within the I band that we have the Z disc, or sometimes it's called the Z line. And the function of the Z disc or the Z line is to attach actin molecules from different sarcomeres. Here's something else that's kind of important to us. So if you find the Z line, find two of them. So here's a Z line here. Here's a Z line here. This identifies the boundaries for a single sarcomere. So we call a sarcomere the area in between the Z lines. Okay, so here's a good opportunity for us to practice what we were talking about. I'm going to identify a part. Feel free to pause it and see if you can identify what each of these parts are. So what is this area that is surrounded by the gray box? That was a dark band. And this is a light band, a dark band, light band, and dark band again. Okay, those were easy. What's this area surrounded by the pink box? Hint, it's the area in between the zigzag lines here and the area in between the zigzag lines here. That's going to be our sarcomere. What's this area here in the green rectangle? Again, that's going to be our H zone. That's surrounding the M line. Ooh, here's a different one. What are we going to call this area right here? Kind of a light yellow box. Well, that is where we see both the myosin myofilaments, the thick myofilaments, overlapping with the thin myofilaments here. So that is obviously our zone of overlap. What about this area in the pink box there? Well, that's going to be our I band or light band that is surrounding the zigzag line inside of it, which is called the Z line or the Z disc. So here again we see this is a cross section through a muscle fiber where they've actually sliced a little of the sarcolemma here, so you can see as that sarcolemma is pulled back, you can see a nucleus a little bit more clearly. Looking inside of the muscle fiber, we can see lots of myofibrils with dark bands and light bands and dark bands, etc. Here they've taken one of these myofibrils and they've pulled it out so that you can see here's a dark band, here's a light band, here's a dark band, here's a light band. They've also included some sarcoplasmic reticulum here because we store calcium there, which we're going to need for muscle contractions coming up in just a little while. This is nice because this is showing that picture that we were looking at before, showing all these myofibrils that are in a muscle fiber. But notice all of the dark bands line up in a row, and all of the light bands line up in a row, and all of the dark bands. So we see dark band and light band and dark band and light band. Now, looking at this photomicrograph, the skeletal muscle under the microscope, you can see dark band and light band and dark band and light band, and that is what causes the striations in our skeletal muscle tissue. All of the dark bands lining up in a row, all of the light bands lining up in a row, and again and again and again. Pretty cool stuff. This particular picture doesn't serve any purpose for the video that we're talking about here, but this picture has been placed into the PowerPoint slide, and the PowerPoint slide is available in Canvas. So here's an opportunity for you if you would like to print up some copies of this and practice identifying the different parts. Being able to identify the different parts of a sarcomere is always something that you could be asked on a test or a final, so you want to be practicing this stuff. All right, and let's start looking at the actin or thin myofilaments and the myosin or thick myofilaments in some greater detail, because it's the qualities of these actin and myosin myofilaments that are going to allow that muscle fiber to contract. So here we see a much better drawing, certainly has a lot more detail than what I was showing before. So here is our M line, that big vertical line right here. Here's myosin and myosin and myosin, and we've got actins in green and a Z line that's connecting the actins from different sarcomeres. When we look more closely, we see that the actin is really a pretty complex arrangement of lots of different proteins. We'll see there's 3 parts. And the myosin myofilaments are going to have these little, they kind of look like a double golf club head sticking up off of them. And it's the interactions between these different parts that are going to allow the muscle fiber to actually get shorter. So you see that the actin, the thin myofilament here, and the myosin myofilament here, where the zone of overlap is, where they're overlapping one another, this is actually going to allow the head of the myosins, which you see right here, are going to be able to pull the actins towards the M line, thus making our muscle fiber, or making our sarcomere, and as a result, our muscle fiber shorter. But let's look at these in greater depth so that we can understand what's going on. So our actin, or thin myofilaments, are made of three different parts. We've got F-actin, tropomyosin, and troponin. So F-actin, the F stands for fibrous actin. So these are two strands or two fibers of individual units called G-actin, and the G stands for globular actin. Now, I like to think as an analogy using Mardi Gras beads here. So if you look, we've got a double strand of Mardi Gras beads that have kind of been twisted over one another. This is really what F-actin or fibrous actin looks like. It's two strands of these bead-like G-actins wound together. So each individual bead is called G-actin. And G-actins have on them binding sites. So particular parts that the head of the myosin is actually able to plug into. And that's going to get our muscle contraction started when that happens. However, we don't want the myosin heads plugging into the G-actins all the time. We don't want a muscle contracted all the time without being able to relax. So let's see what we have here. So we've got two individual strands of Mardi Gras beads. Each bead is a G-actin, or a globular actin. If we take this strand here and this strand here and twist them together, we end up with this double strand, which is our F-actin or fibrous actin. So again, fibrous actin is made up of two strands of G-actins that are twisted together. And here you can see them. So in this case, each G-actin or globular actin is a green bead. And we've got our two strands of G-actins that are twisted together, and that gives us our F-actin or fibrous actin. The next part of our thin or actin myofilaments is called tropomyosin. And tropomyosin is a long rope-like protein that's going to be covering up the binding sites on those individual G-actins. Because remember what I said, we don't want the myosin and the actin bound together all the time, right? Because that would cause our muscles to contract and not relax. So sometimes we're going to need to have those muscle fibers relaxing. And tropomyosin, this long rope-like protein, is going to cover up those binding sites and prevent contractions from happening until we're ready for them. So preventing the premature binding of actin and myosin. But we have to have a switch mechanism. We have to have the ability to move the tropomyosin out of the way sometimes when we actually want the muscle fiber to contract. And that's where troponin comes in. So troponin is going to have three parts, and it's going to be part of our switching mechanism to allow the muscle to contract. The first part of troponin is going to bind to tropomyosin, connecting troponin and tropomyosin together, and we sometimes call that the troponin-tropomyosin complex, especially when we have troponin also binding with the G-actin. So one part of troponin is going to bind to tropomyosin, one part of it's going to bind to G-actin, and that really gives us our troponin-tropomyosin complex. And then one part is going to bind calcium ions. Remember those calcium ions that we were storing in the sarcoplasmic reticulum? When the sarcoplasmic reticulum releases calcium, that calcium is going to go to our troponin. Okay, so here we see our F-actin, our fibrous actin, which is made up of 2 strands of G-actins. Attached to that, we've got this long rope-like tropomyosin here. The tropomyosins are covering up the binding sites on the G-actins. And you can see this a little bit more closely here. The long rope-like tropomyosin is covering up the binding sites on the G-actins. And then here's our troponin, and the troponin is keeping the tropomyosin in place. But sometimes when we want the muscle to contract, we've got to drag the tropomyosin out of the way. And that's why it's important that troponin binds calcium. And we'll get more to that in a little while. Now let's talk about the myosin, or the thick myofilaments. And the myosins are made up of many golf club-shaped molecules called myosin. And here you can see, now I know this is a slightly different picture, but I really like the way this one was drawn. So you can see our actin molecule here, which has got our F-actin, or double strands of G-actins, long rope-like tropomyosins, and in this case they look like kind of pink jelly beans. That would be our troponin. Here is our myosin. So the myosin is made up of many of these golf club shaped myosin molecules that are all bound together. So the 2 parts of the myosin, we've got the tails, and all of those tails are bound together here so that you can see them. And then we've got this area right here, which is called the head. And the head of the myosins have 2 lobes that are going to project out of the tail. And when the head is able to connect with the binding site on each individual G-actin, they're going to form a connection called a cross bridge. And here we see all of the individual pieces together again. So we've got our F-actin, our tropomyosin, and our troponin. And we've got our myosin heads here, myosin heads on the myosin molecule. And then looking over here, we see how they interact in the zone of overlap, that the myosin heads are actually able to plug into the binding sites on the G-actins when we want them to. So remember a little while ago we talked about this concept map. So we went from muscle to fascicle, from fascicle to muscle fiber. Within the muscle fiber, we found sarcoplasmic reticulum, sarcoplasm, and sarcolemma. We're going to modify this a little bit, though, and we'll say that the sarcoplasmic reticulum is going to store calcium, which it does, and the sarcolemma is going to have T-tubules in them, which bring action potentials deep into the muscle fiber. We've got myofibrils, and myofibrils are organized into sarcomeres, and within each sarcomere, we're going to have the myofilaments, actin, and myosin, with actin having F-actin, troponin, and tropomyosin, myosin having the head and the tail. We've finally gotten enough to start to put some of the pieces together. So this brings us to what we call the sliding filament model for a muscle contraction. And the sliding filament model for a muscle contraction explains what happens in a sarcomere in order to allow it to shorten. And when we have all the sarcomeres and all the myofibrils that are getting shorter, that's going to make our muscle fibers and thus our muscles actually contract. And really what's happening here is actin and myosin proteins are going to slide over one another. And by sliding over one another, they're going to make our muscle shorter. And essentially what's going to be happening is it's going to cause the Z lines or the Z discs of each sarcomere to move closer together. And the I bands and the H zones are going to get smaller. And in fact, sometimes the H zone will appear to disappear. And if you'd like to see an example of how this happens, let's do this.
This here is a model of a sarcomere. Sarcomere is a contractile unit for skeletal muscle fibers. This covering, which is acting as a dust cover, but it actually represents the sarcoplasmic reticulum, which is a modified ER, which is storing calcium, which is crucial for muscle contraction. The parts of this you should know, this is the transverse tubule or T-tubule. These are the terminal cisternae off to the sides of that, and they make this triad structure. Okay, so then you can look at this. This is the actual sarcomere. The sarcomere's boundaries are shown at the left and right. This is the Z discs, and they're actually shaped like the letter Z.
Isn't that convenient?
What a sarcomere is creating the striations that you see under a microscope. Now, the striations, these light and dark bands that the muscle has, is a result of these thin and thick myofilaments.
So they're representing the thick or myosin myofilaments in blue, the thin or actin myofilaments in red, and you can even kind of see some of the myosin heads sticking out on the thick or myosin myofilament, and you can kind of see, this isn't the highest quality video, but you can kind of see here's our F-actin, the double strand of G-actins. The tropomyosin is kind of a cream color, and the troponin that's holding them together and has the binding sites for calcium is kind of in green.
So this is a thin myofilament containing mostly actin, and this is the thick myofilament containing myosin. So what happens is the actin's attached to the Z discs, right? You can see that on both sides. The thin myofilament is attached here. The thick myofilament is connected here in the middle. This is called the M line. And during the muscle contraction, the thick filaments, the myosin, walk the actin towards the middle. And it happens on both sides simultaneously, so the whole thing shortens. The Z discs actually get closer together. So what's creating the striated appearance when you look at it under a microscope? When you look at this area here, just this region here will appear light under a microscope. That's called an I band or light band. It appears light because it only has thin myofilaments. This area, from here to here, that will appear dark under a microscope, and that's called an A band or dark band. The reason why it appears dark is because it contains thick myofilaments, and they're thicker, of course. There is a special area right here where there is no overlap with thick and thin. You can see just this middle region where it's just thick myofilaments. That's called the H band, and if you actually look at it,
or H zone,
like the letter H.
Okay, so you get the general idea there. Starting to put all this stuff together. Here is a nice high quality picture showing what we kind of saw in the model. So here's our M line, here's our myosin or thick myofilaments, and here are actin or thin myofilaments here. And here you can see this is what's happening when the muscle fiber or the sarcomere here is not contracting, because you can see the H zone. When an action potential is delivered and the muscle is going to be contracting, the myosin heads actually cause the actins to be moving towards the M line, so much that our H zone you can't even see here anymore. So the actins have actually been pulled, and in this case are touching our M line. So our H zone is completely gone. And this is where we're going to end for part 1. Part 2 of the muscle video is where we're going to be looking at what's actually going on in each of these parts, and we got 2 possible essay questions. So thanks for your hard work. Please make sure you go back and look at the material that we've gone over here, and you really want to be comfortable with the parts before you start looking at part 2 of our video. Thanks, folks. Keep up the good work, and we'll see you around.